So I'm Chris Langdon. I'm a postdoc in the Ingle Lab. I'm also a member of the International Brain Lab. So I'm going to talk to you guys about our efforts to use neuropixel recordings to create sort of a brain-wide picture of neural responses during complex behavior in mice. So just a very brief relevant background for the IBL. So the IBL is a sort of virtual collaboration of about 20 experimental and theoretical labs around the world, trying to work together to understand decision-making in mice. The original sort of motivation, one of the motivations for the IBL was the sort of observation that there's a sort of discrepancy at the level of scales of the sort of scale of complexity of the brain and the sort of scale of efforts in single labs to understand this complexity. So, as a result of this, this sort of piecemeal approach, you have, multiple labs making their own choices about training animals, how they do neural recordings, and then even at the level of analysis of their data, making it really hard to, reproduce results, compare them, etc. So the IBL you can think of as an organization trying to tackle that problem, trying to really build standardized practices all across the experimental pipeline, across labs. Okay, so here's just a nice video summary of a single trial within the IBL task. So on the left-hand side, you see a head-fixed mouse. They're placed in front of a screen where they're showing a stimulus that can either appear on the left or the right side of the screen with varying contrasts, and the mouse needs to indicate its decision about what side the stimulus is on by turning a little wheel to center the stimulus on the screen. On top of this, we experimentally control the probability of the stimulus appearing on either side of the screen in blocks of trials. So we can use this to try to understand how animals use prior beliefs about their environment to inform their choices. So what we also have is multiple camera views of the mouse doing this task. So you can use software like Deep Lab Cut to extract different types of behavior. On the right-hand side, you have sort of the aggregation of all the cells recorded across all the laboratories. So this is after very strict quality control. This is about 35,000 single cells. And what you're seeing here is something like a mean response for each cell on each trial. So reproducibility, we tried to sort of incorporate, along every aspect of the pipeline. So starting all the way from, how we train mice and measure behavior, how we, making sure we have reproducible electrophysiological features from our neuropixels, all the way through to the analysis of this data. So this sort of has culminated in our brain-wide map, and this is what I'm going to focus on for the rest of the talk. Basically, our goal was to use all this data to try to understand how different task-relevant variables like the choice and the stimulus side are encoded in different parts of the brain. All right, so we took a group approach to this problem. What we did was divide into 4 different groups, each led by 1 or 2 PIs and a handful of graduate students and postdocs. Each group was tackling the same problem. How is a given task variable encoded across the brain? But each group could use a different statistical method. So here what I'm showing is results from the decoding group. So what each group tried to produce was a map of, say, an effect size for each relevant variable. So here you have a flat map representation of the brain. So this is a 2D projection of the 3D brain that tries to preserve distances as well as possible. So for instance, this pink area would be midbrain, light blue would be striatum. These light green areas are the motor cortices. These are useful representations because they allow you to see every brain region at once. Okay, so what we wanted was for each group to create one of these maps using a different statistical method. So just give you a feel for these results. White means that we did not do an analysis in that region. Gray means that there was no statistically significant effect due to that variable in the region. Otherwise, it's colored so that the darker regions have the largest effect for that variable. So what you're seeing here is, very large effects for the choice in hindbrain regions. Moving to the right, what we have is the distribution of these decoding results across each region. So you can think of this flat map as arising from the data that generated is this row, this choice row. So zooming in on one region, GRN in the hindbrain. So each black dot would be a decoding accuracy on a given experimental session. So one thing to observe here is that there is some variability across sessions. So the source of that variability can be many things, right? Every time you insert a neuropixel into a region, if there's any topographic organization to that region, slight differences in where that probe is actually hitting the region, that would be one source of variability. Also, this is data across multiple mice, so if there's any subtle differences in behavior. But what we wanted to do is sort of aggregate all these results to create these maps. So for each region, we would take this distribution, say take a mean decoding accuracy, and then also determine whether that was a statistically significant effect after correcting for multiple comparisons across what's like 300 brain regions. Okay, so what we'd like to know is, how much does this picture depend on a choice of statistical method? So for instance, in another group, we looked at a Mann-Whitney test for single cells. So, if you don't believe in population level encoding, you might be interested in just, what's happening for single cells. What do single cells look like? So this approach looked at just whether there was a statistically significant modulation in a neuron's firing rate according to a given variable. And then to get a number for each region, what we would do is consider how many, the fraction of statistically significant neurons. So this is another alternative measure to a decoding accuracy that gives you, again, a picture of how variables are represented across the brain. Okay. So again, we see, at least for the choice variable, again, we see largest effects in the hindbrains. That seems to match our other, our decoder. But okay, so what we'd like to do is, try to extend this idea. How reproducible are these results across statistical methods? Okay, so what we did in total was consider 4 distinct statistical methods. So I talked already about our decoding approach and the Mann-Whitney single cell statistics. What we added to those methods were a metric based on Euclidean distances between trajectories, really the maximum distance between neural trajectories, maximum Euclidean distance, and fitting a general linear model to single cells. So overall, what we had were 2 population level methods and 2 single cell methods. So glancing at this, you do see consistently, large effects in the hindbrain. But overall, what jumps out at you are some differences across methods. We found that the sort of cleanest way to compare all these results is to use a heat map. So on the bottom here, we have each row corresponds to a different statistical method, and each column corresponds to a brain region. So when you display the results like this, commonalities sort of jump out at you as hot places in the map. So indeed, one of the hottest places on the map is these hindbrain regions, picking up our observation that consistently across methods, we seem to see high effects in this region. Some other things to look at, you do see a hot place near the motor part of the superior colliculus and other parts of the midbrain. But overall, the other thing that jumps out is the differences. So if we look at, say, the decoder, you do see large effects in secondary and primary motor cortex and other parts of the isocortex, but not so much in other regions, with other methods. So it seems like the decoder is sensitive enough to pick up an effect in these cortical areas that other methods aren't. And note that this isn't as simple as just using a decoder, just saying that it's the most sensitive method, because indeed you get places, for instance, in the ventral part of the anterior cingulate, where, say, single cell and manifold both reveal an effect, a statistically significant effect, but the decoder does not. So, overall, while, there are some large-scale patterns that you can extract from this data, our sort of view has been that these methods are just, when you have such a large-scale data set like this, any subtle difference in your statistical method can sort of show up somewhere. So you really just need these methods just tell you different things. So what we've tried to do is not present, just an overly simplified narrative of encoding the brain, but try to present the results in as transparent of a way as possible. So just going through some other variables. So again, this was the encoding of choice. We did the exact same thing for other variables. So here we have encoding of the stimulus side. Again, there's sort of large-scale patterns which you can extract. For instance, the stimulus side seems to have a sparser representation across the brain than the animal's choice. As a sanity check, we do see agreement across methods in what you may consider classical visual areas like visual cortex and parts of the thalamus. We do find interesting things that should be explored further. So in hindbrain, again, we find very strong signals, but for stimulus. And as far as we can tell, these are not choice signals. Obviously, stimulus in this task is very well correlated with choice. But we've developed ways to try to tease apart, demix those two effects. And it does seem that there's a sort of, not a transient effect due to stimulus, but a sort of ramping signal due to stimulus in the hindbrain. Last sort of big thing I would want to say about these results. So, one of the interesting things about the IBL task is the experimentally controlled block structure, controlling the probability of stimulus sides. When we try to look for representations of that, we find very little. For instance, the manifold analysis did not yield a single statistically significant region for block. Across decoding in single cell, it's an extremely sparse representation with very little overlaps. However, this does seem to be coming from the fact that mice don't actually compute the exact experimental block, but some approximation of that. And when we're sort of more careful to actually decode what we believe the animals are sort of keeping track of, which would be something like, a running sum of previous choices, it's a slightly more widespread signal. All right, so what I wanted to do to conclude is show you some of the nice web interfaces that the IBL has developed for exploring these results. So in the first link, we have a way of looking at the results of all 4 statistical methods in the IBL dataset. So if you select the project, in this case, the brain-wide map, so we were looking a lot about choice, so we can look at choice, and then you pick the method. Right now we have decoding, and we can look at the effect size. So in each of these different slices, on the right-hand side, you'll have it just colored according to brain region. On the left-hand side, you'll have it colored according to the effect, with the largest effects in the brightest colors. So for instance, here in the hindbrain is GRN, and sweeping through the brain, we do see that that is one of the sort of largest effects due to choice. I think another nice way to use this is to compare it with other methods. So if we were looking at the Mann-Whitney, sort of switching back and forth between these two methods, you can really see the differences in these two methods. Okay, I think a great way to use this tool is in conjunction with another web interface we have for exploring single cells, but with very minimal pre-processing. So say you come here and you see some large effect in GRN and you're suspicious, what you could do is then go just look at single cells in GRN. And what we have is just their raster plots sorted by different trial conditions. So here's a nice cell in GRN where you see it really splitting according to the animal's choice, and peaking right at the time of movement. So I think these two tools together give you a very transparent view of our results. Okay, I think that's it for my time. Thank you to everyone in the IBL, and I'm happy to answer any questions on Slack.